Hello everyone, welcome to our educational webinar titled Bone Marrow, Skeletal Progenitors, and Bone Diseases. My name is Cora Lewis and I'm a product manager at STEMCELL Technologies. I will be your moderator for this webinar. It is now my pleasure to introduce you to our speaker today. Dr. Mara Rimunucci is a professor of pathology at Sapienza University of Rome. She received her MD with board certification in anatomic pathology and a PhD in human pathology from the Sapienza University of Rome and continued her training at the National Institutes of Health, where she received a Fogarty International Fellowship. Mara has a long-standing interest in skeletal biology and genetic skeletal diseases, and has contributed as an expert consultant to the international clinical protocols based at the NIH on fibrous dysplasia. She is a member of the executive board of the International Bone Marrow Adiposity Society and is on the editorial board of Stem Cell Research. Let me now hand you over to Mara to begin the presentation.
Thank you very much for inviting me to give this presentation. Okay, in this talk, I first will briefly summarize the basic properties and functions of human postnatal bone marrow skeletal stem and progenitor cells to remind you why we think that these cells are so important in the establishment, homeostasis, and regeneration, not only of bone, but of the entire bone marrow organ. And then I will give you an example of what happens in the same organ when those properties and functions are compromised, as it occurs in fibrous dysplasia of bone. And I hope that this part of the presentation will be also a good example of how we can use the notion of marrow-skeletal progenitors in human medicine, not only for treating, but also for modeling and understanding human skeletal diseases, starting from the assumption that proper understanding is key to proper treating again. First of all, I would like to remark that all we know on the biology of marrow-skeletal progenitors comes from a sequential series of in vivo transplantation studies at heterotopic sites, and that these in vivo assays still is the only reliable approach that we have to study the biology of these cells and of any other population of putative skeletal stem cells, because this approach is the only experimental assay that allows us to investigate the inherent biological properties and differentiation activity of a given cell type in the absence of any local environmental cue. So in essence, starting from the evidence that boneless fragments of bone marrow transplanted at heterotopic sites regenerate bone first and then establish a newly formed bone marrow cavities, these experiments eventually led to the identification of a small subset of clonogenic marrow fibroblasts that are multipotent and self-renewing. They are multipotent because when their clonal progeny is transplanted at heterotopic site in the subcutaneous tissue of immunocompromised mice, it regenerates all the different human postnatal skeletal tissues, bone, and you can see here human lamin AC positive osteocytes, human adipose marrow, and the human bone marrow microenvironment, which is then colonized by the murine hematopoiesis, thus generating a complete human heterotopic ossicle, a skeletal organoid. And in this skeletal organoid, multipotent colony-forming unit fibroblasts regenerate the compartment in which they reside. In the human bone marrow in situ, colony-forming unit fibroblasts coincide with the so-called adventitial reticular cells. This is a perivascular compartment of the bone marrow stroma that has been identified a long time ago, but has remained poorly characterized for many years because of the very elusive morphology of the cells that can be properly visualized only through special histological techniques, such as, for instance, alkaline phosphatase and confocal microscopy. As you can see here, the branched and really elusive morphology of these cells. We now know that adventitious reticular cells express some antigens that allows us to distinguish colony-forming unit marrow fibroblasts from their non-clonogenic stromal counterpart, and that they are regenerated in the heterotopic ossicles produced by multipotent colony-forming unit cells. So this regeneration is the evidence that marrow skeletal multipotent progenitors are genuine skeletal stem cells, and they have been renamed skeletal stem cells. This association of marrow skeletal stem cells with the blood vessel wall is not surprising, since microvascular pericytes have been implicated with progenitor functions in many human postnatal tissues. However, for marrow skeletal stem and progenitor cells, this association is not merely anatomical. Maybe we could say that this is a strategic position from which these cells interact with and organize neighbor cells and tissues. Histological studies performed in the heterotopic ossicles at different developmental points, in fact, have shown that the interaction of human self-renewing colony-forming unit fibroblasts in the heterotopic ossicles is very important for the establishment of a marrow sinusoidal network and for the establishment of an hematopoietic microenvironment through which human, self-renewed human colony-forming fibroblasts, meaning skeletal stem cells, contribute through cell-cell contacts. You can see here a human CD146 positive cells interacting with a mouse megakaryocytes, and through the secretion of hematopoietic cytokines, such as, for example, angiopoietin-1, as you can see here by immunolocalization, and many other molecules, some of which are shown here in this transcriptomic analysis. So in other words, the self-renewal of the human skeletal progenitors, human skeletal stem cells in the heterotopic ossicles coincides with the establishment of a functional marrow sinusoidal microenvironment. And this occurs during bone development, during our postnatal life, and is recapitulated in this experimental system. Furthermore, at perivascular site, marrow skeletal stem cells generate the postnatal differentiated skeletal phenotype, osteogenic cells and marrow adipocytes. And the importance of perivascular adipogenesis should not be overlooked, because this is another way, another mechanism through which marrow skeletal stem cells regulate the activity within the bone marrow microenvironment. Because when adipocytes are formed at the blood vessel wall, and so they increase in size at the blood vessel wall, sinusoids collapse and they are excluded from the circulation, although they retain an intact endothelial layer. When adipocytes undergo lipolysis, sinusoids dilate and the blood flow resumes. And in this way, marrow skeletal progenitors and stem cells indirectly regulate the amount of distribution of blood flow within the marrow cavity. And on a large scale, we can say that they regulate the conversion of adipose marrow into hematopoietic marrow and vice versa. Now, before continuing with marrow skeletal stem and progenitor cells, I would like to mention an issue that is dominating the skeletal field nowadays, whether or not these properties and functions are duplicated in other populations of mesodermal-derived progenitor cells. Because this is the concept, the idea on which the notion of mesenchymal stem cells has been based. As you know, this notion reads that mesenchymal stem cells represent a ubiquitous system of equivalent progenitors throughout our body. As a matter of fact, CD146 positive perivascular stromal cells can be identified in different compartments of our body. For example, in the skeleton, they can be identified in bone marrow and in periosteum, or they can be identified in other non-skeletal tissues, such as for example, skeletal muscles, adipose tissue, and many others. So in vivo, these cells all share the same anatomic location, and in vitro, they share the same surface phenotype and the same clonogenic activity. However, when they are transplanted in vivo with an osteoconductive carrier, they behave in a completely different way. Skeletal cells, other than bone marrow-derived progenitors, generate bone, but do not establish a bone marrow microenvironment. Non-skeletal derived cells regularly fail to generate bone and bone marrow, although some of them may regenerate tissue-specific phenotypes in appropriate experimental systems. For example, CD146 cells isolated from skeletal muscles are able to generate myotubes when transplanted in the mouse at heterotopic sites with a non-osteoconductive carrier, such as growth factor-reduced collagen, matrigel collagen, and in other experimental systems. And this different behavior is clearly reflected in the transcriptomic profile of these cells. Their transcriptomic profiles are perfectly segregated, and we can find a bone signature profile only in skeletal-derived cells, bone marrow and periosteal cells, and in the production of hematopoietic cytokines only in bone marrow-derived stem cells. However, it must be specified here that this does not imply that marrow-skeletal stem cells are the only type of skeletal stem cells in our body. For example, we have reported on a population of skeletogenic cells that can be isolated from the cord blood. These perinatal CD146 positive clonogenic fibroblasts are able to form bone when transplanted with the osteoconductive carrier, although in contrast with the marrow-derived progenitors, they are also able to generate cartilage. They have an inherent chondrogenic activity, whereas they have a very low, if any, adipogenic activity. And other groups have also reported on different population of skeletal stem cells. For example, there are many many manuscripts. I'm just mentioning this beautiful work by Chen and colleagues that was published last year, in which the author have identified a population of skeletal stem cells in our prenatal and postnatal life, skeletal stem cells that generate cartilage, bone, and hematopoietic stroma. And there are, as I said, many other reports on different types of skeletal progenitors. So altogether, these data demonstrate that rather than a system of mesenchymal, equivalent mesenchymal progenitors, different cores of human skeletal stem cells exist in our body, different time and space. And this is in perfect agreement with the physiology of our skeleton. In our skeleton, in contrast, for example, with the hematopoietic system, the differentiated phenotypes are generated in an asynchronous manner. Cartilage is produced very early during development, but it's not normally generated in the postnatal life within the bone marrow cavity. In contrast, adipocytes are not generated in the prenatal life, and they only appear during the postnatal life during growth. So this kind of flexibility, this asynchronous appearance of different phenotypes likely requires different populations of skeletal stem cells with slightly different properties. Going back to marrow skeletal progenitors, obviously their complex activity must be perfectly regulated, and many different pathways and molecules and factors have been involved in this regulation. I do not have the time to mention all of them. I just would like to remind you the parathyroid hormone, because the importance of this pathway, the PTH-PTHRP pathway, has been demonstrated in many studies on transgenic models and in human, in human bone tissues. For example, this is a transgenic model that we had the opportunity to study, to investigate many years ago. The model was generated by another group, and it's characterized by the constitutive, by the expression of a constitutively active PTH-PTHR1 receptor, PTHRP, sorry, receptor in osteogenic cells. And as you can see here, these mice are characterized by excess bone formation in the prospective marrow cavity during growth, with a delay in the establishment of the hematopoietic end of the adipose marrow. And this is another transgenic model recently published by Fan and colleagues, in which the PTH-PTHRP receptor has been genetically deleted in mesenchymal progenitors expressing the PRX1 promoter. And these mice are characterized by osteopenia and expansion of the adipose marrow. And the importance of the PTH pathway on the adipocyte generation has been confirmed by this author also on human samples. And so this data clearly indicate that the PTH-PTHRP pathway is a major regulator of the bone marrow organogenesis and therefore of the function and differentiation of marrow skeletal stem and progenitor cells. And since this pathway uses the GS-alpha cyclic AMP system as the most important, the major downstream mediator, this data directly brings us to the disease that I would like to talk about, fibrous dysplasia of bone, a disease caused by mutations of the gene encoding the alpha subunit of the stimulatory G protein, GS-alpha. These mutations occur during embryonic development, likely due to an abnormal methylation process, and thus they generate a somatic mosaicism. And they are gain-of-function mutations, and thus they induce abnormal increase of intracellular cyclic AMP in mutated cells. Fibrous dysplasia may occur as a mono, poly, or even a panostotic disorder, and due to the somatic mosaicism in many patients, it associates with non-skeletal diseases, mainly endocrine hyperfunction and skin hyperpigmentation in the classic McCune-Albright syndrome, but also with muscle myxomas in the so-called Mazabro syndrome, or also liver and pancreas abnormalities in particular cases of the McCune-Albright syndrome with very early onset in the neonatal age. However, in all the different clinical contexts, except for these cases with very early presentation, fibrous dysplasia usually represents the most severe and less treatable expression of the mutated genotype due to recurrent fractures, pain, bone deformities, and also nerve compression when it involves craniofacial bones. And this is because in fibrous dysplasia, the normal bone and bone marrow are replaced by a pathological, mechanical unsound fibro-osseous tissue. So hematopoiesis and hematopoietic supportive stroma disappear, adipocyte disappears, and the marrow cavity are filled in by this fibrotic osteogenic tissue, within which pathological bone trabeculae are formed. These bone trabeculae have a woven structure, abnormally oriented collagen fibers. They are hypomineralized and are abnormally resorbed. And so many years ago, based on these tissue changes, we hypothesized that fibrous dysplasia results from the abnormal differentiation in function of GS-alpha mutated skeletal stem and progenitor cells. And mutated cells that are not able to generate any more adipocytes and to establish a correct bone marrow microenvironment. And this hypothesis is supported by the fact that GS-alpha mutated colony-forming unit fibroblasts can be isolated from the fibrodysplastic tissue, in which, interestingly, they are always associated with wild-type CFUF, meaning that the lesion itself is a mosaic, is a somatic mosaic. And when these GS-alpha mutated colony-forming units are transplanted at heterotopic sites along with their normal counterpart, they regenerate a fibrodysplastic ossicles. And I would like you to note here that this is an example of how a human genetic disease can be transferred into immunocompromised mice by using postnatal skeletal stem cells. But the role of marrow GS-alpha mutated marrow skeletal stem and progenitor cells in fibrous dysplasia has been clearly confirmed by GS-alpha transgenic models. These are models that we have generated in our lab. And in this model, the GS-alpha R201C mutated sequence, one of the two mutations that generate fibrous dysplasia, is expressed in the entire skeletal stem cell system, meaning skeletal progenitors and differentiated osteogenic cells. Because in these mice, the transgene, the expression of the transgene is driven by the ubiquitous and constitutive promoters elongation factor 1 alpha. And as you can see here, these mice reproduce a disease, a skeletal disease that mimics human fibrous dysplasia with fibro-osseous lesion, hypomineralized bone, abnormally resorbed bone trabeculae. In contrast, mice in which the same transgene, the same mutated GS-alpha sequence is expressed under the control of an osteoblastic promoter, in other words, mice in which the expression of the transgene is restricted to differentiated osteogenic cells, do not develop fibrous dysplasia. These mice develop a different skeletal phenotype, an high bone mass phenotype, characterized by excess bone. As you can see here, this bone is normal, is fully mineralized, and is not associated with a fibrotic marrow. So these models confirm that in order to develop fibrous dysplasia, in order to reproduce fibrous dysplasia, we need mutated osteoprogenitor cells expressing the mutated GS-alpha sequence. And so investigating fibrous dysplasia as a stem cell disease over the years have been extremely productive, because we have been able to explain some aspects of the natural history of this disease that could not be reasonably explained based on the biology of differentiated osteogenic cells. For example, it is known that in old FD patients, skeletal lesions often become quiescent, and we have observed that this associates with a reduction in the number of mutated CFUF within the lesion. And in some patients, this in turn is followed by the regeneration of a normal bone and bone marrow microenvironment by the intralesional wild-type skeletal stem cells. In other words, this is a kind of natural experiments of cell therapy that is spontaneously made in some of these patients. On the other side, by studying fibrous dysplasia, we have gained very important insights into the physiology of our skeleton. For example, it was by studying skeletal stem cells and their progeny fibrous dysplasia that we have recognized for the first time that these cells produce the phosphaturic hormone fibroblast growth factor 23 that of course explain the hypomineralization of the FD bone, but it also reveals an endocrine function of our skeleton, the ability of our skeleton to regulate the mineralization of the bone matrix through the regulation of the activity of the kidney. But obviously, the most important question that we need to address is what are the molecular mechanisms that link the expression of the mutation in osteoprogenitor cells, in skeletal stem cells and their progeny, to the development of fibrous dysplasia lesion, to the tissue change that characterize fibrous dysplasia lesion. Identifying this mechanism is very important to identify potential target for a medical therapy for the disease. And I would like to remark here that fibrous dysplasia does not have a cure at this time. And so to try to address this point, we are using in parallel an in vitro model of the disease generated by using normal skeletal stem cells and our transgenic mouse models. This in vitro model of fibrous dysplasia has been generated by transducing skeletal progenitors and stem cells from normal donors with a GS-alpha R201C mutated sequence, with a lentiviral vector carrying this mutated sequence. The idea here was to generate a large amount of mutated skeletal stem cells and progenitor cells. And obviously, it is very difficult to obtain these cells from FD patients because it's a rare disease, because in many cases the diagnosis is made through clinical analysis and imaging techniques that do not have biopsies and so on and so forth. And so we wanted to generate this model to better study the molecular abnormalities of mutated skeletal stem cells. So colony-forming unit fibroblasts from normal donors have been transduced, and as you can see here, they express the transgene, and they also express the minimum molecular phenotype of fibrous dysplasia, meaning increased intracellular cyclic AMP. In vitro assays show abnormal differentiation of these cells, whereas transcriptomic profile shows dysregulated expression of many genes. And among these dysregulated genes, in particular among these, the upregulated genes, there are two genes that are very important for the clinical morbidity of the disease: the matrix-GLA protein and the rank ligand. The matrix-GLA protein is a local inhibitor of bone matrix, of extracellular matrix mineralization at large. So it is not produced by normal bone-forming cells, by normal osteogenic cells. Its expression, as you can see here, is upregulated in transduced skeletal progenitors. And these results is confirmed in spontaneously mutated skeletal stem cells isolated from FD patients and also in FD tissue. So matrix-GLA protein, we have identified this molecule as a mediator along with FGF23 of the hypomineralization as a cause for the hypomineralization of the extracellular bone matrix in FD lesions. Rank ligand is the most powerful osteoclastogenic factor that we know, and it is upregulated, as I said, in transduced cells, and this result has been confirmed recently by other groups on human tissue samples and also on in different transgenic mouse models that they have been generated, different transgenic models of the disease. We have confirmed the upregulation of both these molecules in our models too, and matrix glyprotein is overexpressed in the fibrotic tissue and in bone-forming mutated osteoblastic cells, and the same is for rank ligand that is upregulated within the F deletions, mouse F deletions. And we recently focused on this molecule, on rank ligand, because a humanized anti-rank ligand antibody already exists for human therapy. Denosumab, it's the denosumab that has been approved by FDA for some skeletal diseases, such for example, osteoporosis and the giant cell tumors of bone. And a murine equivalent of these antibodies is also available. And so this has allowed us to test the effect of rank ligand inhibitions in our mouse models. And these are the results. Mice have been treated with the anti-mouse rank ligand antibody twice a week for 14 weeks. As you can see in this radiographic analysis at different time point, the administration of the antibody induced a reversion of the skeletal phenotype of these mice and a progressive increase in bone density. This occurred in the tail, in the femurs, and in all other involved skeletal segments, whereas the disease, as expected, steadily progressed in control animals. These radiographic changes are due to the fact that the fibrous dysplastic tissue following rank ligand inhibition is replaced by newly formed bone. And it's very important to note that this newly formed bone is detected in skeletal segments that were affected at the beginning of the treatment, but not in skeletal segments that were free of the disease at the beginning of the treatment. These unaffected skeletal segments maintained their normal shape, a normal bone marrow cavity, a normal bone marrow architecture. We only observed, as expected, an increase in the thickness of the metaphyseal cartilage due to the inhibition of bone resorption. And it was even more interesting to note that this newly formed bone was, at least in part, lamellar, had a lamellar structure, and most important, it was fully mineralized, meaning that this data have been published a few days ago, and I mean, so they are very, very recent. So we have still to investigate the mechanism through which the rank ligand inhibition also reverts the hypomalacia, the osteomalacia observed in these mice. But certainly they demonstrate that rank ligand inhibition can be a valuable therapeutic option for FD patients. As I said, we need further studies. We need to understand the mechanism for these tissue changes, and we also need to solve some problems. For example, the rebound of the disease following the discontinuation of the treatment. But still, we think that these results are really, really promising. This was my last slide. I hope that this has been a significant example of how, by studying the basic properties of skeletal stem cells, by studying the performance, the characteristic of skeletal stem cells in skeletal disease, it is possible to identify specific pathogenetic mechanism and as in this case, to identify potential therapeutic approaches. And I would like to thank all the people that over the years contributed to this work from many different labs all around the world. I do not have the time to list all of them, so I decide to acknowledge in particular Professor Paolo Bianco for his outstanding contribution to the skeletal stem cell field and to the understanding of fibrous dysplasia of bone. Thank you very much.
Thank you very much, Mara. That concludes the presentation segment of the webinar. We will now begin our Q&A session. Is it possible to reconstitute the bone marrow compartment of myeloablated mice using the bone marrow that colonizes the ossicles formed by the heterotopic transplantation of bone marrow-derived CFUFs?
Okay, we did not, this is a very good question. We did not perform this experiment, but the answer is yes, probably it's possible, because an hematopoietic niche is re-established in the heterotopic ossicle.
Are there currently any approved therapies for fibrodysplasia? If so, what do they involve?
The only therapeutic approach currently available for fibrous dysplasia is the use of bisphosphonates, meaning other molecules able to inhibit bone resorption. And obviously there is surgery in particular cases, corrective surgery when it is required, but there are no specific therapies for the disease.
What is your opinion on the therapeutic potential of MSCs, or stromal progenitor cells, and how they are currently being applied in clinical trials?
If we think about bone regeneration, mesenchymal, quote unquote, mesenchymal stem cells isolated from bone marrow certainly can be an option, although we have to keep in mind that the bone has a very complex structure, and so it's not easy to reproduce this structure in the lab or following injection of skeletal stem cells. As for the other clinical trials and proposed clinical applications, I think that most of them are not, at least many of them, are not based on rigorous preclinical data on suitable experimental system. We now know for sure that mesenchymal stem cells are able to regenerate phenotypes from the tissue of origin. They are not able to generate other types of phenotype. So I think that before clinical trials, we really need suitable in vivo experimental system.
How do the rigors of the ISCT criteria in trying to define stromal progenitor cells affect your work?
We do not follow any specific procedure. We use clonogenic culture, meaning very low density cultures. We only work on bone marrow derived skeletal progenitors, and we know that some of those clonogenic cells are multipotent. Obviously, there are no antigens, or there is no way to identify which one of them is multipotent before in vivo transplantation.
Okay, I think that's all the time we have for today. Thank you, Mara, for your time and this excellent talk. Goodbye.